I'm gonna record on my screen. Uh-oh, somebody's unmuted. We're good now. Okay, so we're gonna try to keep this nice and short so that we're not wasting anyone's time here. But I thought since yesterday was, or is it today? Today's the first day of spring, right? Or was it yesterday? Yesterday was the first day of spring. So we're gonna do something with plants, right? We're all also kind of in the module right now about living organisms, plants, bacteria, protists, all that stuff, right? So we're gonna review designing a controlled experiment, grow plants, and also hopefully not kill them, but some of them may die. And don't ask me for advice because I kill every plant. So when we're designing an experiment, right, we need a question. Sometimes we need background research or a purpose. We need a hypothesis, right, a super important component with a reason, not just, I think my plant will grow. Really, we want to know why. We need variables, we need a design and a procedure, we need data and a graph, and then we need results and an analysis. So today I'm going to be helping you guys from your question that you pick. I'm going to be helping you with your variables and your procedure / design. But basically you're going to be kind of left to your own devices here in terms of designing your experiment. Because part of this is going to be you figuring out how to design this. And it can be an editing process, right? You can send me an email, you can submit documents to me, say, hey, does this look right? Is this a good experiment? And I'll try to give you some tips on it, but it can be complicated, right? Especially you'll see once you start trying to collect data on these plants. So in case you haven't viewed it yet, there is a biology lab report template, which I'm going to be posting this PowerPoint, by the way. If you click on it, you're brought to this page where you will have to make a copy of the document. It doesn't let you edit in it. I want to let me zoom in. There we go. Okay. So this lab report template is what we're going to be aiming to have by the end of the experiment. It has all the basic same stuff that I just went through. It also has additional stuff like safety, right, materials and equipment. And it has instructions on how to do a graph on the computer if you need instructions for that. There's also a bunch of websites I found out that just graph for you, which is pretty cool. So feel free to use those. But regardless, we're going to be using this lab template. So make sure you have opened it and viewed it. All right, so first things first, you were supposed to pick a question, right? I actually added one additional question because I thought it could be a cool one as well. But we're gonna have, how does the type of soil affect the growth of radish plants, type of liquid, amount of sunlight, amount of water, or acid rain, which is similar to type of liquid. I see people saying stuff in the chat here. I want to let me click on chat. Okay. So, yes, it says no content on the, yes. Thank you, Zach, for saying exactly what I do. Yeah, so guys, you'll notice on all of these labs, there's not usually content until the day of, because they're supposed to be labs that we do together, right? So I don't want to post stuff in advance because then you could have already done this without me, right? And the point is we're going to do it together. So these are the question choices, right? You're going to be choosing which question you want to explore, which is also interesting because at the end of this, we're going to have data from a lot of different questions, right? Assuming that we all pick a different one. We have radish seeds and garden bean seeds, right? So you can choose which kind of seeds you would like. If you're doing this from home, you can choose your own seeds, but I recommend something like bean sprouts, radishes. These things, they sprout quickly. You don't want to be waiting 3 weeks for your plant to sprout, right? Because that's a lot of wasted time. You can also use already grown plants. as long as they're the same plant and they're the same size. We'll talk about why that's important in a few minutes. So there's a lot of options here. In fact, you could leave today and decide that you want to do your own plants. That's fine. If you're watching from home and you need plants, right, I'm going to have the supplies here for basically the next month. So if you need to come in and grab supplies, let me know. If yes, you're going to grow plants at home if you're not here, because people will be taking these plants home. By people, I mean everyone that's here. So, oh my gosh, this chat. This is hard to talk and do the chat at the same time. Okay, so we are going to now talk about how to design this. So hopefully you have a question in mind, which question you would like to know. We're going to talk about variables. Every year, you probably have noticed in science, you learn about variables, and it never seems to end, and somebody's not muted. Who's not muted? Okay. So every year you probably learn about variables, right? I will say it took me probably 4 years of science in order to be able to actually understand variables, right? And part of it is when you try to design your own experiment, it really starts to click why these are important. So our independent variable is something that we as scientists are changing. So in this case, depending on your question, this could be the type of liquid, this could be the type of soil, this could be the amount of water, this could be acid rain. Okay. Part of the fun of Zoom, crazy echoes going on when people have their headphones in, they're like, okay. Right, so we need to be very clear. We're only changing one thing. If you start changing 2 or 3 things, you're not actually going to be able to get results. Think about it this way. If you have 2 radish plants, and one of them we're watering. Okay, I thought maybe someone was unmuted. Make sure, guys, when you come in that you're muting yourself so we don't hear your background noise. We have 2 radish plants, right? And we decide we want to change the amount of water and the amount of sunlight. Terrible experiment design, right? Because then if both of our plants die, or one of them dies, we don't know if it's because of the water or the sun, right? There's no way for us to tell which one of those things killed the plant or prevented the plant from growing or made it turn a weird color or whatever it did. So we only want to change one thing, and that's what becomes hard. We have to keep everything else constant, which is what we call control variables. There's more than one of them. We have to keep everything else constant. So if we're going to change the amount of light, we're going to grow some plants in a closet, grow some outside. Pretty cool experiment. Kind of surprising results, right? We want to keep the water amount the same. We want to try to use the same pots, or in this case, cups. We want to use the same type of seeds, right? We want to give them the same amount of attention, because there is actually research that talking to plants helps them. Can anyone suggest the scientific reason behind that? What do you breathe out when you're talking? Carbon dioxide, right? And what do plants use carbon dioxide for? To make oxygen to go through photosynthesis, right? So if you're talking to your plant in the closet more than your one not in the closet, it could be a problem. So we want to think about those things that we're going to keep the same, right? And then we have to think of something to measure. So what kind of stuff could we measure on a plant? The height, right? That's a really great indicator of health, right? If the plant's growing, it's getting taller, it's getting bigger, right? We can assume that it's thriving. We could also measure color, right? Which is a little hard to measure because you're going to end up having to create a table of colors and use those colors. But we can think that, right? We can look at a plant and say, this plant doesn't look healthy. If it's like yellow, if it's wilting, right, if it's brown, you can tell from the color that it's not doing well. You can also measure something like the number of leaves. We can also do something like the number of flowers, right? I didn't pick flowering plants because they take longer to sprout usually. They will flower eventually, but they're not mainly for flowering, if that makes sense. But there's a lot of different things we can measure. Height, I would say, is probably the best and easiest dependent variable for any of these experiments because it's easy to record, right? And you'll see a clear difference too. You can tell, and sometimes plants might grow and then they might fall over because they get too tall, right? Which is another thing we're gonna have to think about. We're also gonna have a control group, which Zach in the chat said, are we doing a control plant? Yes, we are, right? Because we need something to compare it to, and that's what control groups are for. It's a normal group. What do plants normally do, right? Normally they're in a window, they're getting water, right? They're in soil. We want to have a normal plant. I say normal in quotations because are any plants really normal? I don't know. But we want to have a plant that we can compare it to. Because if you just start growing plants and watering them with Gatorade and they're thriving, it doesn't mean anything if you don't have a regular plant to compare it to. Right? People are going to be like, oh, cool, what does the plant normally look like? I don't know. Okay, your experiment's not good then. You need to be able to say, the regular plant in normal plant conditions, right, grew this tall, but the Gatorade plant grew 3 times as tall. So the variables are going to be super important in the design, right? These are all things we have to think about before. Part of the problem with science experiments generally is that this stuff's already told to you, right? They already pick the variables for you. They already tell you what they are, and you just glimpse through it, and it doesn't matter. But there's a lot of thought that has to go into these, right? So we're going to do an example here of how does acid rain, oh, that's right, I can do that annotate thing. How does acid rain affect plants is the question that I'm going to model, right? You can think about this for your own question as well. So if I'm doing how does acid rain affect plants, what is my independent variable? What am I changing in this experiment? What am I doing to the plants? Exactly. I'm using acid rain or the type of liquid. Now you may be thinking to yourself, how do you make acid rain? You just take a little vinegar, mix it with some water, and you have acid rain, right? Not too complicated. Real rain outside is technically probably slightly acidic, right? But we'll make a super acidic rain for this. You don't want just a mildly acidic rain. So acid rain is going to be what we're changing as scientists. What are we going to measure here, right? We kind of already talked about this. We might want to measure the height here, right? Or some people said they might want to do color, right? That's fine. I'm going to pick height. I'm going to be seeing my acid rain plant versus my regular plant, which one grows taller. My control variables, what am I going to have to keep the same? What am I going to have to keep the same? The type of plant, right? Some plants are actually can do pretty well in acidic conditions, right? It doesn't bother them. In that case, it wouldn't be good to compare a plant that's doing well in acid rain to one that's dying, right? So the type of plant, super important. What about the amount of liquid? What if I'm dumping acid rain on one plant and just giving a little spritz of water to the other one, right? Doesn't make sense. These things are like common sense kind of, so they're hard to express, right? But we want to be thinking about the amount of liquid that we're giving the plant. Other ideas for control variables. What about where we put the plants? We want to have the same location, right? The only time the location would be different is if you're doing sunlight versus darkness, right? Because then obviously to have your plant in the dark, it's going to have to be in a different place than the sunlight one. All right, and then our control group. For this one, it's pretty self-explanatory. We're just going to have a plant that's getting regular water, right? So now the complexity of this experiment is going to be up to you. To do this acid rain experiment, I could do something as simple as taking 2 cups, filling them with soil, putting in, let's say I want to use radish seeds, putting in 3 seeds in each cup, maybe just in case some of them don't grow. Labeling them, right? Acid rain, regular, A, B, whatever, if you remember what that means, A for acid rain, B for not acid rain. And then you're going to decide how often are you going to water them, right? Are you going to water them every day? Are you going to water them every 3 days? Do you need to set up a reminder in your phone because you suck at watering plants, right? How often are you going to record the data? Every time you water, every day, right? These are all decisions that you're going to make. Now, you could make this even more complicated. If you're super interested in science and you really want to go in on this experiment, right? You could have multiple levels of acid rain. So you could start adding more cups here where you can actually have maybe a pH of, let's say a pH of 4, right? Which I can help you make that with vinegar if you'd like. You could have a pH of 5, pH of 6, and then pH of 7, which is regular water. So you could differentiate it by pH. So then you would be looking at super acidic rain versus maybe just a little bit acidic rain. Right? So that's another way that you can make this more complicated. Same with the other experiments, the light and dark one. For example, you can have one completely in the dark, one in the sunlight if it's sunny, and one maybe inside, right? Where it's kind of getting light, but not as much light as outside. Right, so there's a lot of different ways here. I want you to have a minimum of 2 cups, obviously, because we need a control plant and a plant that you're manipulating. You're welcome to do as many plants as you would like. Now, type of liquid, right, is a great one. You could pick any kinds of liquids you would like. You could use soda, you could use Gatorade, which is actually a pretty interesting one to use. You could do like vitamin water versus regular water, right? So there's a lot of options there for type of liquid that you would want to use. Make sure there's like a rationale behind it. Why would watering a plant with a certain liquid maybe be beneficial to the plant? Or maybe it won't affect it, right? So like for Gatorade, a lot of people have argued in the past, has electrolytes in it, right? That might actually help the plant. Does it? If you do the Gatorade one, get ready for some smells. That's all I'll say. If you really want to annoy someone in your house, just get a cup of soil, pour a lot of Gatorade in it, and just leave it there for a while. It ferments, and then it smells like vomit. Okay, so you're going to be doing this process for your question, right? And you might choose to do the acid rain one, that's fine. Just making sure that you're thinking about here your variables, how many different plants you want, right? In fact, it helps me to draw this out, where I literally would draw the different cups I'm going to have, what I'm going to label them, what I'm going to do to those cups. So this could be an experiment where once a week you're watering your plant. It could be something where every day you're interacting with the plants. So okay. So designing your procedure, could you do lemon juice for acid rain? Yes, right? Lemon juice is another great way to make something acidic, right? In fact, that might be better than vinegar. Maybe it will not hurt the plant as much, right? That's up to you. Okay, so again, I kind of already started talking about this stuff, but are you gonna water every day? How much are you gonna water? Right, that's something to think about too. Don't go crazy with the watering, right? Some people are like, oh, I'm gonna water 10 milliliters of water, right, which sounds small, but in this small of a cup, right, that's quite a bit of water. So you could even do a spray bottle. You could do 5 sprays of water, right? So don't do too much water or you'll just have a swimming plant. How often are you going to record your data, right? And where are you going to record it? Are you making a Google Doc for this? Are you doing this in a notebook, right? How and when? The other thing about the data is if we're planting 3 seeds in this cup, we potentially could have 3 plants, right? Are you going to do an average of those plants? Are you going to measure each one? Right, so thinking about that as well. How much data are you going to collect? You could do it either way. As long as you're being clear in your write-up, I took the average of all the plants in my cup, right? Or I measured each individual one, however you want to do it. But keep that in mind. Don't put 10 seeds in a cup because then you're going to be measuring 10 plants in one cup. You're going to need at least 2 weeks of data, right? That doesn't mean you need to collect every day in that 2 weeks, but I want your plants to experience their variable for at least 2 weeks. If you're only collecting data once a week, I would increase that time. If you do acid, can you base it on the pH? Yes, I think that's what you should do. So you are probably wondering, wow, this sounds like it's going to take a lot of time. Yes, and you'll have noticed too that in Canvas, a lot of you guys already have this lab due, right? It might have been due last week, it might be due today-ish, it might be due next week, right? I'm not concerned about that due date, and you shouldn't be concerned either. This experiment could take you a month, right? It could take you 2 weeks, that's fine. When we get into May, if you still haven't completed your experiment, I'm going to start putting in zeros for that, right? Because that means by then you'll have had lots of time to do this. So this I kind of want to be something that's going on in the background while you're doing your regular coursework. So don't worry about turning anything in yet. In fact, I'll see if I'll try to change the due date, but for those of you who weren't with us last semester, Canvas hates me. And every time that I created assignments last semester, it deleted them, which was fun. So it's not the best teacher-friendly system. But I can try to change the due date. Otherwise, you can always submit a work in progress, right? What you've done so far, just so that it goes off your to-do list, right? And then I can leave you feedback that way. So you're going to need at least 2 weeks of data here, really, to get a good idea. Right, and then you're going to be filling in that lab template. So I'm here to guide you if you need help, but I'm not going to force help on you. So if you get stuck on something, you can ask me. Okay, so how much should you water it each day? I would say I would probably water every other day, and I would probably only do like, I don't even know, maybe like 3 tablespoons of water at most, right? Probably less than that even. That's going to be up to you. I don't know the magic number. Also, I kill plants. So maybe you should do 1 tablespoon of water. Okay. And then can you use an already grown plant? Yes. So if you go to any store and you want to pick some plants to use, right, just make sure that they're the same type of plant and that they're roughly the same size. Obviously, they're not going to be identical, right? But you want to make sure they're close in size. Measure them in the beginning so that you know where they're starting. Where do you get the supplies? Well, if you're here, you have supplies right here, right? I'm going to keep the supplies available here, so you're welcome to come by MIWA and pick up seeds, cups, soil. I can even let you check out a graduated cylinder if you would like to do that. We don't have a ton of them, but a few people may be able to check those out. So you can either buy your own supplies, right? Or you can email me to find out when I'm here, which is Tuesdays and Fridays, all day. And you can come by and pick up supplies. So where do we get the supplies? Yep. So make sure that you're thinking about that, right? If you want to get your own plants, if you already have plants at home that you want to use, right, that's fine. Shouldn't be expensive, right? A whole thing of radish seeds costs $1.79, and we're using red Solo party cups, which are pretty cheap as well. You can puncture holes in the bottom of these too to help with the drainage. And then soil, right? You could buy soil, or if you have your own yard, right, you might be able to take soil from your yard even. Okay, so the next time that we do a virtual session, we're going to be kind of checking in about this. Most people will probably be finishing up, or at least pretty well into their experiment, so we can kind of start talking about the results, right? We can talk about what happened to the plants. We can even start talking about the soil, right? What's in the soil, what's maybe missing from the soil, are things to think about as well. So, what do you turn in, right? I also kind of said this already. Is acidic the high pH? It's the low pH, Zach. Okay, so the full lab report write-up is what you're going to be turning in. And again, you can do drafts of this, right? Once you turn it in, it's not over. I'll give you feedback, right? If you choose to not do anything to your lab report, then obviously you're over with it or done with it. Having a hard time talking today. Then you're done with it. Otherwise, you can continue to revise it, right? Just like with any assignment, you can always go back, resubmit it. You can email me, text me, right? Turn in something if you want me to look at it. I'm happy to. For those of us who are here right now and are going to be getting started on the plants, right? I'm happy to talk through your experiment with you if you already kind of have ideas about what you're going to do. So that's pretty much it. People who are at home, right? Do you have any questions before I end the Zoom meeting and we get to planting here in person? How many plants should you use? Minimum of 2. I think for best results, you want to have 3 to 4. Yeah, I think 4 is a good number, Zach. What time am I at school on Fridays? From 9:30 to 2:30, because the building closes at 2:30. Yep, I'm here from 9:30 to 2:30, right? If you can't come on a Tuesday or a Friday and you can come on another day, let me know and I can try to find a teacher that's here who can help you, right? I don't know why I'm talking to you guys, you're all here. You guys, I can find someone to help you if you can't come Tuesday or Friday. Tuesdays, I'm here from 9:30 to 3:30. I usually have 15 meetings a day on Tuesdays, but I'm happy to leave the meeting to help you, so let me know. And then Fridays, I'll be here 9:30 to 2:30. We do have a middle school lab Friday mornings, so if you come in and see 40 middle schoolers, do not be scared. I'm here and I will get you your plant stuff. All right, any other questions from people at home? Thursday, Devin, yes, I can leave it with people at the front desk for you, and I'll leave a little note that it's for you so that you can grab it, okay? All right, anybody else, if you have you need something on a certain day or whatever, just send me an email or send me a text and we will get it solved. All right, otherwise, I'm gonna stop sharing. We're gonna end the meeting, and happy planting.